The overall goal of this procedure is to analyze the expression of microRNA in microglia. This is accomplished by first dissecting the brain and spinal cord and then homogenizing the central nervous system or CNS tissue. In the second step, mononuclear cells from the CNS are isolated by a Perkal gradient, and then the microglial population is identified by FACS. Next, the microglial RNA is isolated, and the quality of the sample is analyzed. Finally, real-time PCR for the particular microRNA of interest is performed. Ultimately, the delta-delta CT method can be used to show the relative level of expression of a particular microRNA in microglia.
Generally, individuals new to this method will struggle because they fail to isolate sufficient number of mononuclear cells from the CNS or because the sample is contaminated with myelin and cell debris. Both cases result in low-quality RNA preparation with either low concentration of RNA or contamination with protein respectively. Demonstrating the procedure will be me and Tatiana Veremeyka, a postdoctoral fellow from our laboratory.
After perfusion of all the mice, dissect the brains and spinal cords. Then, place one brain and one spinal cord at a time into a 15 milliliter Dounce homogenizer, adding 5 milliliters of PBS to the tissues each time. After performing 10 to 20 gentle strikes, pass the homogenates through 70 micron cell strainers into individual 50 milliliter conical tubes and centrifuge the tubes for 5 to 7 minutes at 834 times G and 4 degrees Celsius. After discarding the supernatant, resuspend the brain and spinal cord homogenates from 2 to 3 mice in 5 to 7 milliliters of 70% Perkal. Then overlay 7 milliliters of 40% Perkal and spin the Perkal and cell solution for 30 minutes at 850 times G, 20 degrees Celsius with no break. Discard the damaged cells and myelin debris at the top of the 70% Perkal. Then collect the mononuclear cells at the interface between the 70% and 40% Perkal solutions and dilute the collected cells in PBS at least at a 1:1 ratio. After spinning down the cells, resuspend the pellet in half a milliliter of PBS and transfer them into a 1.7 milliliter Eppendorf tube. Begin by transferring 50 microliters of the density gradient sorted cells into a new 1.7 milliliter Eppendorf tube and add 350 microliters of FACS buffer to the tube. Then, after adding 5 to 10 microliters of anti-FcR antibodies to the tube, incubate the cells for 10 to 15 minutes on ice. Split the cell sample into 2 Eppendorf tubes. To one tube, add 1 microliter of anti-CD11b and 2 microliters of anti-CD45 antibodies, incubating the cells for 10 to 15 more minutes on ice. Label the second tube negative control and place it on ice. After incubation with staining antibodies, add 1 milliliter of FACS buffer to both tubes and spin down the cells in a microcentrifuge for 5 minutes at 1200 times G. Then fix the pellets in 400 microliters of 1% paraformaldehyde in PBS and vortex the tubes to prevent clumping. Finally, analyze the cells by flow cytometry using the negative control cell sample and BD compubeads. Begin by subcutaneously immunizing a group of 5 to 10, 8 to 12-week-old C57 black 6 mice with 150 milligrams of MOG 35 to 55 peptide in 4 milligrams per milliliter of complete Freund's adjuvant to induce EAE. Then administer pertussis toxin intraperitoneally on day 0 and on day 2 post-immunization. After isolating and staining the cells as just shown, Resuspend the pellets in 400 microliters of PBS instead of fixative just prior to sorting. Then, fill two 1.7 milliliter Eppendorf tubes with 300 microliters of PBS supplemented with 10% FBS for collecting the samples. Finally, sort the microglia and macrophage populations to a high purity using the gating strategy illustrated in this representative density plot. After sorting, reanalyze the microglia and macrophage populations to determine the purities of the isolated cell samples. After spinning down the sorted cell populations at 1200 times G for 5 to 7 minutes in a microcentrifuge, carefully remove the supernatant and freeze the cell pellets on dry ice. Next, transfer the frozen cell pellets to regular ice for thawing. Add 300 microliters of lysis buffer from a Nirvana kit, and then gently mix the cell solution by pipetting 5 to 7 times. Then add 30 microliters of the homogeneous additive, vortex the cell suspension for 30 seconds, and then place the tubes on ice for 10 minutes. Now add 300 microliters of the acid phenol chloroform and vortex the tube for 30 more seconds. Then, after spinning down the cells for 5 minutes at 9500 times G, carefully transfer the upper 300 microliters of the supernatant containing the aqueous phase to a new 1.7 milliliter Eppendorf tube. Mix the solution with 375 microliters of 100% ethanol. Transfer the entire tube contents to a filter cartridge from the Nirvana kit and spin the cartridge for 1 minute at 9500 times G. Next, discard the flow-through buffer. Add 700 microliters of wash solution 1 from the Nirvana kit and spin for another minute at 9500 times G. After washing the cartridge 2 more times with 500 microliters of wash solution 2 from the Nirvana kit, elute the RNA from the cartridge with 60 microliters of preheated nuclease-free water. Finally, use a nanodrop spectrophotometer to assess the purity and quantity of the RNA. After performing real-time qRT-PCR to detect microRNA in the microglia and macrophage samples, PRT-PCR curves for fluorescently labeled miR-124 and SNORNA55 PCR products can be created as shown here. In the first graph, typical curves for the amount of fluorescently labeled specific PCR products for control SNORNA55 RNA samples isolated from microglia or bone marrow-derived macrophages, as compared to the number of PCR cycles, are shown. Note how the CT values for analysis of the SNORNA55 expression from the different cell populations are determined at the intersection of the baseline with the lower part of the linear range of the qRT-PCR curves, as indicated in the graph by the green and blue vertical lines. In this second graph, the amount of fluorescently labeled specific PCR products for the experimental miR-124 RNA samples from microglia or bone marrow-derived macrophages compared to the number of PCR cycles is shown. Note how the CT values for analysis of the miR-124 expression from the different cell populations are again determined at the intersection of the baseline with the lower part of the linear range of the qRT-PCR curves, as indicated in the graph by the green and blue vertical lines. After determining the CT values for each of the samples, as for the SNORNA55 and miR-124 expression in microglia and BMDMs from normal and EAE mice shown in this table, subtract the CT value for the control SNORNA55 sample from the CT value for the experimental miR-124 sample to calculate the delta CT values for each duplicate of each sample. Then subtract the delta CT of the reference sample from the delta CT values of the other samples to calculate the delta-delta CT values. Finally, the relative level of expression for each sample is calculated as 2 to the power of minus delta delta CT. As seen in this density plot, in the case of a good isolation of microglia from normal CNS, 95 to 98% of the cells will represent CD11b positive, CD45 low microglia, with 2 to 5% of the cell suspension being made up of CD11b positive, CD45 high perivascular macrophages. Less than 1% of CD11b negative, CD45 high lymphocytes, or CD11b negative, CD45 negative astroglial or oligodendroglial cells or cell fragments will be present. Comparatively, in a bad preparation, there is a significant contamination of CD11b negative, CD45 negative cells or cell fragments, such as astroglia or myelin particles, making the cells unsuitable for RNA isolation and further analysis. In addition, CD11b negative, CD45 high cells also may be present, indicating contamination by blood lymphocytes due to insufficient perfusion. In this density plot, the three populations of the cells present in the CNS during neuroinflammation, CD11b positive, CD45 low microglia, CD11b positive, CD45 high macrophages, and CD11b negative, CD45 high lymphocytes are shown. In this figure, the quality of RNA, as determined by gel electrophoresis after a good versus bad isolation, is shown. When the RNA is isolated in a clean manner, an RNA ladder and ribosomal RNA are evident. When the isolation process is performed poorly, smear and low molecular weight degradation products are evident. These final data demonstrate expression of miR-124 in normal adult microglia, microglia of mice with EAE, and microglia isolated from mice during early stages of development. In this first bar graph, the relative expression of miR-124 is much greater in microglia from normal CNS compared to that observed for bone marrow-derived macrophages. Similarly, although there is an increase in miR-124 RNA in macrophages from the CNS of mice with EAE, microglia still express 5 times the amount of miR-124 RNA. This final bar graph demonstrates how changes in the level of expression of miR-124 in microglia during the development of mice of 1, 2, and 4 weeks of age, as compared to adult 8-week-old mice, also can be evaluated using this method.
While attempting the procedure, it is important to remember to have a sufficient quantity and good quality of isolated cells, which will guarantee good results for the entire experiment.